Yeah.
I'm going to start slowly. I'll start by introducing myself. My name's Tom Curtis. I'm the Professor of Environmental Engineering in Newcastle University. And my role really was that I proposed that we should have an N2O working group. And I'm very pleased to see so many people joining. And I think it's worth taking a moment at this point to just remind ourselves why we're here. I think this is one of the reasons why we're here. This is one of the Icelandic ice caps, which I had the pleasure of going up last week for my holidays. It's extremely beautiful. But as we went towards it, we saw something which I think was in many ways rather less beautiful. So this is a glacier, as you probably recognize. And this is the snout of a glacier on the left here. And for those, and this is in rather less attractive weather. And if you look at that right-hand photograph, in the distance in the mist, you'll see the dirty grey moraine-ridden ice of the snout of a glacier. And there, my good friend Ingvar slugging up with a very large rucksack, and ropes and all that kind of malarkey. But I want to draw your attention to the rock in the foreground. Just there. And on the right-hand side, I've got a picture of what's written on that rock. And it says the 1st of August, 2022. That's where that glacier was on the 1st of August, 2022. And that's where that glacier is now. It's retreated, as you can see, quite a long way in just 8 months. And these are 8 winter months where we don't think the glacier perhaps should be retreating as quickly as it is. And this is just one of the many, many, many canaries in the global coal mine, if you pardon the expression, which we're seeing right now, and which has been foreseen for at least 30 years. That we're looking at a fundamental shift in the climate of the planet unless we do something pretty serious about it. And of course, there's a lot more at stake here than my holidays. There's it's existential really for life on the planet as we know it. However, I am very pleased and proud that the UK water industry has stepped up boldly and ambitiously to create a roadmap for net zero for the water industry. This is great leadership for the industry, but honestly, without being too hyperbolic, I think it's great leadership for the world, because every country in the world needs water and sanitation, and we all want to deliver that with as little carbon impact as possible. But this is not to underestimate the scale of the challenge that we're facing, particularly in the water industry, where we have very long-lived assets. You know, when we pour concrete for a wastewater treatment plant, we expect it to last for 50 years or even more. But that's why we have such a great opportunity in EBNet to contribute to this great challenge, because the bacteria, we can change those in 50 days. So there is a huge potential, I think, through a joint collaboration, thinking and challenging of each other in this community to make real change on the kind of timescales that we've already heard we need from Greta Thunberg. You know, we need solutions fast and cheap, and Ms. Thunberg gets a lot of good and bad press, But actually, I think at heart, she's an optimist. She says, and I agree, that we can actually do something about this, but we have to do it pretty quickly and pretty smartly. So why an EBNet group on N2O? Well, clearly it's a big problem. And I think in the UK, we have not only the problem, but some sort of a special UK versions of the problem. For example, we make particular use of biological filters here, trickling filters, which are I think very understudied. We have many carbonaceous activated sludge plants, which are very understudied. There is, as I'll be saying, huge biological potential, which I think is largely untapped. But also, I think maybe to create a focus for the many small initiatives which are taking place. Many water companies are sitting up and worrying about nitrous oxide, and they're doing perhaps what water companies do when they're not quite sure what to do, and that is to get a PhD student to go and have a look at it. And so there's many small initiatives like that. We'd like to get people to come together and to talk and to challenge and sometimes to agree. But also, I think will hopefully be a showcase for new talent. Now, I don't want to disrespect the fantastic old talent, like, for example, Tom Stevenson, who's been looking at nitrous oxide for many years. But there are also many really interesting, super young people on the block, and we're going to hear from one of them, Carlos, later on this afternoon. And hopefully, if you're listening in and you think, oh, I might be new talent, hopefully we'll create a focus for you. And I know the EBNet group are particularly keen on promoting and encouraging early career researchers. So nitrous oxide, I think, is I think ultimately a biological problem with ultimately biological solutions. Now, I know there are non-biological sources of nitrous oxide, but they are not, in the context of the water industry, the biggest problem, or indeed likely to be very often the biggest solution. We're going to hear a little bit about this from Carlos and others subsequently. But just to give you a very sort of quick reminder. If you're a person of a certain age, you may be taught that this was your nitrogen cycle back 20 or 30 years ago. And unfortunately, for many people, it still is what they think is going on in wastewater treatment plants. But there have been revolutionary advances in our understanding of what's going on in the nitrogen cycle in the past 20 years, both of our understanding of what's doing it and our understanding of how to study them. So we definitely understand now that the nitrogen cycle is a great deal more complicated than we thought before. And I certainly don't have time to go through all these slides, but it's annotated with some of the steps and, very importantly, with some of the critical genes. And with the advent of really fast, cheap gene sequencing technology, we're able to monitor and understand many of these steps in a way that we couldn't have thought possible 40 years ago. And here's just an example. On your right here, we have a picture of some ammonia oxidizers. They've had their ribosomes labeled with fluorescent gene probes. The bluey purpley one are the ammonia oxidizers that convert ammonia to nitrite, and they're surrounded by the yellowy greeny ones. Those are nitrite oxidizers that are converting the nitrite to nitrate. And we also know, because my colleague Ben Allen has sorted out these cells and sequenced them, that these are also surrounded by denitrifiers, which are taking the nitrate and the nitrous oxide and turning it into nitrogen gas. And on the left-hand side, we see the relationship between the ammonia loading on a cell in picomoles per cell and the nitrous oxide produced by those cells in an activated sludge plant here in the northeast in County Durham. And I'm not going to go into that except to point to something else we know. So here's the numbers of ammonia oxidizers. The load on the ammonia oxidizers in the Severn Trent region in different activated sludge plants, again in picomoles per cell. And we can see here straight away that there are order of magnitude differences in the load on the different cells. So perhaps order of magnitude differences in the nitrous oxide emissions from these treatment plants. And we also know, and I won't explain how, we can look at, we know that not only the numbers, we can know, and this is actually quite old work now, by looking at the DNA and the RNA expression, which genes are switching on in which treatment plant when. I'm not going to bore you with the details. This work is over 15 years old, but it shows the ammonia oxidizers switching on as they go down an oxygen gradient in one loop activated sludge plant. So I think we have, although we have a big problem, we also have a very big opportunity for both the industry and for researchers. In the short term, I think we can do great work measuring microbes and measuring emissions. And I think that as well as maybe managing our emissions better, we'll probably end up with more efficient treatment plants with lower failure rates and more rational use of the very important resources we use to run them. I think in the medium term, we can certainly see artificial intelligence being used for sort of self-driving sewage work. So we actually are able to use the sequencing technology in something like real time to make sure we operate at just the right balance between failure, emissions and energy use. And I can also foresee as having what you might call intelligent bioaugmentation, perhaps to reduce emissions in certain places. But in the long term, we're going to go for net zero by design, and we will get a new suite of technologies for wastewater treatment that are going to meet the new realities of the climate change era. And I'd be really like to think that some of the people listening in, especially some of the early career researchers listening in, are actually going to make a big contribution to this. Now, if you want to participate in the EBNet group on nitrous oxide, then you can write to EBNet at ebnet.ac.uk. I think you're probably able to get that down quite quickly, and they'll make sure that the group gets and collates all the responses, and hopefully will include you in further events. And now I've already spoken too long, and I just want to give you the agenda for the rest of the meeting. Well, you start off with an introduction by me, which has already got on too long, as I've said, and now we're going to go into a talk by Carlos Domingo-Felez from Glasgow University, and Carlos is originally from Galicia and came out of the very fine Environmental Biotechnology group in Santiago de Compostela, but then he spent many years in the Danish Technical University working on nitrous oxide emissions, and has quite recently been very fortunate that he's come to the UK and is now working in the outstanding group led by Bill Sloan in Glasgow. And we're going to follow that up with lessons learned from Denmark after Carlos has finished speaking. We're going to have a little time for questions and answers afterwards, and when the questions are over, we can draw the meeting to a close.
Yeah, thanks, Tom. So I'm Carlos, lecturer at the University of Glasgow, and now today I want to give a overview on how N2O is produced, different methods that exist for quantification, how it's currently being reported, and like some details on like both models that are available up to date. So what I'll focus today is mainly on the scope 1 of emissions. So we'll put the boundary on the wastewater treatment plant.
Hang on,
we'll stay on kind of the scope 1, so direct emissions. It's important to note that in wastewater treatment, carbon footprint, you can see here for different plants, it was Denmark and Sweden. A lot of the carbon footprint is driven by direct methane or N2O emissions. So a lot of the carbon footprint, it's basically from greenhouse gas emissions. So it's really important to tackle this problem if we want to make some wastewater treatment plants not even energy neutral, but also carbon neutral. They can account from 20 to 50% of the carbon footprints that were studied. And the important part is that the carbon footprint is very sensitive to greenhouse gas emissions. So a single 1% on the N2O emission factor, we might sound very small, can change the carbon footprint by 50 or 80%. So they're like really important in terms of the carbon footprint. Here you can see in another study, it was a Danish water work, but in this case you can see like over the years in blue, there are the direct N2O emissions. So even if the plant has been going in net-wise, it's going towards almost zero because it's producing electricity and it's becoming more carbon neutral. If we remove the N2O emissions, that would be a huge part and it would become negative. So it would be quite positive effect. So over a long year, this has been a field that has been studied for some years now already. And I think in general, what has been done, the approach was starting for about 20, 30 years ago, insights and discoveries, lab scale, for microbial production, chemical pathways if there were, and then moving into reactor, trying to validate and test those hypotheses, then developing models to test and understand the mechanisms behind. And now we're at the point where there's already a lot of observations and already some control strategies for N2O emissions in full-scale plants. So this is kind of an ongoing and a circle that fits to each other. But yeah, we're still understanding mechanisms, still proving them in the lab, but already implementing some strategies in full scale. So how it is produced? Important part is during water treatment. It's mainly biological nitrogen removal. So this is going to be the key process. If we look at it, very typical nitrification and denitrification pathways, we have some N2O being produced during nitrification and some N2O being produced during denitrification. Now, the fact that it's produced doesn't mean that it's stripped. And that's the problem when it gets stripped. If N2O came in the influent and it's not stripped in the anaerobic reactor because there's no aeration, there's no stripping, it's not a problem. The problem becomes when it goes to the aerobic zone and it might be stripped. So high concentrations are not always, but it's a huge risk of N2O. So we could see that probably the influent doesn't have a big N2O coming in, and effluents typically don't have high N2O going out. But any N2O that lives in the effluent, because there's going to be very clean on solids, is going to be directly probably stripped or discharged and probably emitted in the recirculation line, for example. Here is not going to be a problem because it's going to stay back in the anaerobic zone. So yeah, just keep in mind, one thing is where it's produced and another one is where it could be stripped. We know that N2O depends on different variables, and you probably have, if you're familiar, you probably know they depend on nitrite concentration. High nitrite typically leads to high N2O, DO, pH, the carbon to nitrogen ratio, the type of COD that you have in the system, temperature, and all of those, of course, drive what type of microbial community you have. So it's not going to be the same, community that is mainly full of heterotrophic bacteria like activated sludge, which has a small fraction of ammonia oxidizers, or a partial nitration reactor, for example, where it could be a high amount of AOPs and a small amount of nitrifiers. So it's quite complex process. Now, based on literature and what we've seen from the microbiological studies, this is how we typically understand that it's produced, where we have no need to look at the details, but there's some autotrophic pathways, heterotrophic pathways, and chemical reactions. So ammonia oxidizers, denitrifiers, and some chemical reactions. And the key point is that when we measure, all of them get mixed and together. So when we measure N2O, we measure the mixture of all these pathways, and we don't know which one is participating more. Now, let's look into detail what Tom was showing before. This is the updated kind of nitrogen cycle, but I'll just show the ones that are relevant for N2O. These are the autotrophs, but Anamox is not going to be relevant. They can produce intermediates, so they might affect secondary ways, but they're not relevant. One pathway that is called, or we call it nitrifier nitrification, is related to hydroxylamine. And hydroxylamine is a very reactive species that comes from the oxidation of ammonia. Another pathway comes from, it's called nitrifier denitrification. And when ammonia oxidizers are exposed to high concentration of nitrite and there's not much oxygen, then they tend to reduce nitrite into N2O. And of course, during denitrification, N2O is also an obligate intermediate of denitrification. So you can see that for the blue lines for autotrophs and AOBs, N2O is a byproduct, so it's not necessary always for growth, but for denitrifiers, it's an obligate intermediate. So it's always going to go through in their pathway. And there's some abiotic reactions, but what we've studied, we've realized that it's typically only very acidic systems where hydroxyamine and nitrite are very high. So it's not typically relevant in wastewater treatment operations at normal pH values. But yeah, we focus, if you look at N2O now, so the conclusion here is that we have 3 pathways that are producing. It's going to be mixed in the liquid, and 1 pathway that can consume N2O. So the nitrifiers are the only ones who can consume. Now, if you're not familiar, hydroxylamine and nitric oxide are typically not monitored in any wastewater treatment plant and almost not in the labs. So the true substrates for N2O are typically not monitored, which means that we monitor secondary substrates, could be like nitrite, ammonium, nitrate, but we don't really look at the real substrates. So that's important. This would be we can monitor them, but they're more complicated and there are not so many sensors available. Now, of course, every wastewater treatment plant is different. And for example, even within AOBs, those blue arrows that I was showing, AOB species do not respond equally to environmental conditions. So different, depending on the species, the rates between intermediates and how they produce N2O is different. And for heterotrophic denitrifiers, they also show a high diversity, and they have this truncated metabolism where heterotrophs are just a mixture of species, which some of them are lacking certain genes. So for example, this species would have the first 3 steps and it would produce a lot of N2O. It would only produce N2O. But if we had this species, for example, it would be a pure N2O consumer. So it can only grow on N2O. And this one in the bottom, that would be what we typically call complete denitrifier. And you can see, for example, in the figures below, the range of N2O concentrations that are observed in wastewater treatment plants. And there's a big range of concentrations. So we have from very low milligram to very low microgram range, which means that there's a high diversity of N2O reducing bacteria that are thriving in these different conditions. So on the right side, you can see different N2O consumers, and their growth curve typically means that, some of them are adapted to low concentrations, and all the ones are adapted to higher concentrations. So overall, every plant has a unique microbiome, which means that pathways are not going to be always the same, and the plant will not respond to N2O the same way to different environmental factors. As an example, if we only look at ammonium oxidation, then we only look at nitrification and nitrifier denitrification. So for example, like partial nitritation reactor where AOVs are very abundant. You can see here on the figure, left side, N2O production rate, and on the x-axis, the ammonia removal rate. The higher the ammonia removal rate per cell, the more N2O they tend to produce. So it's very similar to the figure that Tom had shown before, where basically when cells are stressed, they might produce more N2O. So in this case, we're measuring the combination of both pathways compared to the ammonium they are removed. When you look at it from the plant point of view, the amount of ammonium that gets removed is the same. So you don't really see a 1% change or 2% in N2O emissions. You don't really see it in your nitrogen balance, but it's just being emitted as N2O. But these pathways, we can actually calculate what's the contribution of each one in different conditions. So there are, we could add stable isotopes, typically more in the lab because they're very expensive and more complicated to work. And we can track specific atoms that are labeled, radio-labeled, and then we can see what pathway they are following. So we could investigate whether this N2O is coming from the dark blue pathway or the light blue pathway. You can also label nitrite, so you can label substrates to know where they're coming from. Or other option is the natural abundance of isotopes or site preference, because naturally there's these isotopes where there's one nitrogen that has a heavier weight, and depending on where they are, we can actually predict which pathway is being formed. So for example, there's been a recent study where they could take 4 activated sludge, where was the contribution of each pathway? And in their case, N2O was mainly produced by heterotrophic denitrification, and nitrified denitrification and hydroxymonoxidation were not so important. So even at a full scale, there are ways of quantifying how much each pathway is contributing to the total. Because again, we typically always measure total N2O. So yeah, keep in mind that 3 pathways produce it and 1 consume it, and they might be always happening at the same time, which we just don't know the relative contribution of each one. There's a nice IWA report that was released in last year, like very short ago, where it has some chapters that are very interesting. So if anyone's interested, they can check also mechanisms, factors affecting N2O emissions, and how to quantify N2O and methane from urban water systems. One of the key things, I think this wraps it pretty well. If you want to measure N2O emissions from a wastewater treatment plant, first you have to decide type of methodology you want to follow, sampling strategy, analysis, and the data. I will just focus on key points that I haven't heard in other webinars. And for example, to quantify the emissions of a whole plant, the mobile tracer dispersion method. In this case, a tracer gas is released next to the wastewater treatment plant. Down the wind creates a flume. And then on the other side, you can measure both the tracer and your pollutant. And then you can calculate how much of the pollutant is being released compared to your tracer, which is a known amount. Of course, it's a bit more complicated than online measurements, but in this way, you can also pick up on specific maybe hotspots that you didn't know your plant was emitting. And typically for methane, they could picture or they could figure out where you would have hotspots of methane emissions that were not expected. And for unit scale, typically you could be for full scale or lab scale, there are liquid and gas sensors available. So could be a hood or a liquid sensor. We will hear more. So for a point source, in this case, we just monitor a small surface or a point. Basically, in a flux chamber, we can have a tracer gas, or typically in the aeration chamber, for example, we would have the gas that transfers N2O into the atmosphere. And the flow meter, we know how much air is being transferred through that surface. We can measure the concentration of nitrogen N2O gas, and then we can calculate the mass flow of N2O that's being emitted. You can see this hood that can be transported and move around any open surface reactor. But we can also use a liquid sensor. In this case, it could be placed same way that you have DO sensor, ammonium nitrate sensors. So there's some benefits of gas phase and there's some benefits of liquid phase. With gas phase, we have all these measurements. We are measuring the flow rate because we will know. We are measuring and the concentration of N2O in the gas phase is also quantified. So the rate of emissions, we already know all the measurements. The problem is during non-aerated phases or non-aerated reactors, we cannot always use these hood measurements to calculate the N2O emissions. But the benefit again is that we have direct measurements of the emissions. On the other hand, with liquid phase, we can track dynamics regardless of aeration or non-aeration. So it's very good to understand mechanisms of how the N2O is being produced, but we need a mass transfer coefficient that relates the concentration of N2O in the liquid phase and the emissions that are transferred into the gas phase. There are methods that exist where you can use to calculate the scalar coefficient, but we are adding a bit of error or approximation to the emissions. So these are going to be direct calculations, and these are going to be based on your KLA values. Now, full-scale reactors. In this case, for example, we placed 4 different sensors throughout the lane, non-activated sludge phase alternating system. You can see during one day almost that the 4 sensors were giving quite similar signal. So there was no spatial heterogeneity within the reactor, which means that one sensor could be located here, and then we could track and assume that all the concentrations in the reactor are the same. In this case, could be a point source again. So a hood or a liquid sensor would be enough. I want to mention that there's already existing protocols, and because there's a large body of knowledge from many long-term campaigns that have been done in the past with different duration, in different locations, with number of probes, and specifically like Switzerland, Denmark, and Netherlands, they have a wide experience on these campaigns. There are some plants like tricking filters, some other biofilm systems, and carbon issues that have been less monitored. So that's where there's more room for improvement. But yeah, just for people who are interested, there's already protocols of how people have done their measurements. So how do you calculate or what do you need to calculate N2O emissions? The rate, if we consider grams of N2O nitrogen per day, we need to measure concentration of N2O in the liquid phase and a mass transfer coefficient. The rest of the parameters are flow rate, volume of reactor, so this should be known. Now, let's go through ideal conditions to maybe more real conditions. If you consider small reactor, ideal conditions, everything is perfectly mixed. We would have one transfer coefficient that is representative of the whole reactor and one concentration of N2O that is representative of the whole reactor. But if we move towards maybe bottom aerated reactors, the aeration, so the mass transfer could be just one number, but there might be a profile of N2O concentrations while it's moving through the lane. If there's some gradients in oxygen concentration, there might be some gradients in N2O. And there's a gradient in ammonium concentration, there might also be gradients of N2O. If we look at surface reactors, then of course the KLA is going to have at least 1D profile, because we're going to have a lot of emissions next to the rotor and no emissions down the water lane. But it could be that the reactor is very well mixed, and then one concentration of N2O is representative of the whole system. And kind of the worst case scenario would be where you have a surface aerator, where there's also some non-aerated zones or poor mixing, where you might have even a 2D map of emissions and also a 2D map of concentrations. So some examples, for example, here on the right, this was a small plant where oxygen was very high next to the rotors, and as it was moving along, the DO was decreasing, but it also had an anoxic zone in the middle. So in this case, there could be some spatial heterogeneity, and one sensor might not be representative to catch the true concentration in the reactor. In this case, where they were feeding part of the influent and the RAS into an anoxic zone, the water was flowing this way, and then because they were feeding the other half of the influent down the line, this was also diluting the biomass. And diluting the biomass with influent meant that they were loading more ammonium per cell. And as we saw before, if you load more ammonium per cell, that might be that AOBs start producing more N2O. So in this case, depending on how they were feeding, they were making spatial heterogeneity based on the distribution of the influent and the RAS, which actually they could manage to decrease by changing the strategy. So how many sensors do you need, or what would you need to monitor? This is important to check if the system is pretty well mixed and you know your DO and ammonium concentrations are mapped and they are constant, then probably one N2O sensor could be enough. But this is actually a good point where if you see gradients of N2O, it probably means that your reactor is not very well mixed or not completely mixed. So you might even find dead zones or some issues that might affect the overall performance of the plant. In that case that I will show before, with this hood, we could measure gas concentrations in front of the reactor. And with that, we could calculate a KLA value that was not just 1 point, but like a map of KLA values in front of the rotor. So with this, we can, so we move the hood in different points in front of the reactor. We measure airflow velocities, and with that, we can calculate KLA values. And with that, we can calculate the total emission of the system. And as you can imagine, for example, here in front of the rotor, this area represented 90% of the emissions. So in these systems, only the concentrations in front of the rotor are going to be important because that's where all the stripping is happening. And you can see in red, that's where we first placed the sensor because that's where all the DO and ammonium the plant had located. So we had them there. But then we realized that just in front of the rotor was the key position for us to place the sensor because that's where all this sampling and stripping was occurring. Sorry. So yeah, N2O liquid concentrations could vary on the surface of the reactor and could be due to poor mixing or depending on the spatial design of the reactor. And that's going to have a direct effect on the predicted emissions. So if you look at the equation, your N2O liquid concentration has a direct effect. Here you can see we had 3 sensors located in that previous reactor that I showed. And maybe you cannot see them here because the shape of the histograms look similar, but the values were different. They were proportionally different. And here you can see on the right the correlation between the signals of the 3 sensors. So they're highly correlated, so they were just proportional, but they were not the same. But depending on which value you take, you might overestimate or underestimate the emissions. At the bottom, you can see in green ammonium concentrations, in red nitrate, and then DO is in blue. This is a phase alternating, so it aerates and stops aerating every kind of hour or 2 hours. And you can see how ammonium goes down, goes up, goes down, goes up. And in black, the 3 N2O sensors, liquid sensors. Here, when ammonium starts being loaded much higher rates, well, in this case, even the aerators cannot even keep up and they cannot aerate to normal set points. But the N2, you can see in black how they start going up. So the 3 sensors they're responding similarly. It's just like the proportionality they are different. But in this case, this is a small plant, and maybe the mixing was not as good in like bigger ones. But this happens also in other places. That it could be that not only you have variability spatially, but also temporal. So how long do you have to measure N2O? I can say, like, to understand dynamics and mechanisms, shorter campaigns can give you a good idea. And maybe with a small investment, you can get a good idea of where you are and what type of mechanisms could be happening. But to predict the emissions of your system, you need seasonal variations. So months to year to capture. At the bottom, you can see, for example, this campaign in SBR in Switzerland, where they did for several weeks. If you miss this last week, you're going to miss probably the highest emission of the period. And here you can see on the right 2 lanes, also in Switzerland, where they had different months over 1 year. And you can see that certain months, they have a significant higher emission rate than other months. So if you just measure from September to December, you would be highly underestimating the N2O emissions of the system. So to get an accurate representation, you need over like at least a 1 year campaign. Because if not, you risk of probably underestimating emissions, but you could also overestimate if you catch one of these peaks in the emissions. So aerobic granular sludge, same problem, high variability. You could see in an SBR where we have certain cycles that have huge emissions and other cycles that are much lower over several months. And also depending on the hour of the day, there was no correlation between the cycles. So there's a very high variability of N2O in general. So there's a huge values for standard deviations divided by means. So the variabilities are pretty high. Why is that happening? Because N2O is way more dynamic than ammonium or nitrate, and the relative changes within this short time can be really high. Ammonium and nitrate, have you seen the previous figure? They vary with the daily load, but they don't have so significant changes as N2O. N2O has peaks that could change orders of magnitude within minutes. And because these emissions are highly variable, this happens also one day within the hour or in long campaigns. So if we see an histogram here of estimated emissions per day, you can see that most of the emissions have a low value. But then we have some days where we have huge emissions of N2O. And this is what we saw also in our reactor, where we had most of the days we have low emissions, but then we have some periods where we have very high concentrations of emissions. And as you can see from this distribution, N2O emissions, they're not described by normal distributions. And I imagine them more as kind of flow rate distributions, where you would have like log normal with long tails. So you can imagine this, that you have most of the days dry weather, and then you could have some rain events that increase, in this case, the emissions. For example, in this phase alternating activated sludge, we saw that 50% of the emissions were happening only with 11% of the points. And in an MABR with activated sludge, with 23% of the points, they were responsible for 50% of the emissions. So how to report? The key figures that are used for reporting are typically total or kilograms of N2O nitrogen per year, would also be converted to CO2 equivalents. And the important figure here is the emission factor, where we calculate the N2O that's been emitted divided by the total nitrogen that has been removed, or how much nitrogen has been loaded into the plant. And it could also be converted into milligrams of N2O per person equivalent per day. But typically we talk about emission factors. So think about N2O that's emitted divided by nitrogen or ammonium removed. There's already a lot of correlations are trying to be found with IPCC data or with national campaigns, where we see trying to find correlations between N2O emissions on the left and nitrogen loading to the plant on the right. Also here, the N2O emission factor with the nitrogen removal. So typically, plants that operate quite well and they have high nitrogen removal, they tend to emit low N2O. Now, one of the issues I think with using these correlations now in long term is that after you stop a monitoring campaign, you probably want to use it to mitigate and decrease the emission factor, which means that these emission factors that are reported are probably already being changed, so are a bit dynamic. So probably everyone who's been monitoring has implemented some strategy to decrease. So if they monitor again, any of these points might be going down. So just keep in mind that this is going to be some kind of dynamic values. If we look by processes, the emission factor, and then if it's activated sludge, oxidation ditch, SBRs, there's no really a pattern between them, and there's a high variability also within each process. And if you look also geographically, there's not a significant pattern between them. But one thing that I always find that it's missing is that the variability of each system is not accounted for. So all these points are not accounted for with the internal variability. So we've seen that this system has a very high variability, but when we're moving now into these first reports, we only consider average values. So I think that's not going to be very representative, and in the future we might be able to use more information, which we already have. I'll briefly describe that there's this document that they released in Denmark in 2020, where they had a very extensive campaign and studies, in this case 9 plants. And even I think it's in Danish. I don't know if it's translated to English, but it's very easy to translate nowadays. But you can see the figures that where they report all the numbers of different plants, loadings, emission factors. And what they've seen is that even if the IPCC in 2019 updated the value for an emission factor of 1.6%, they've seen that in the study they've done, that for them is closer to 0.84% of nitrogen emitted per nitrogen load. So these are the numbers that are starting to come up that might not always be representative of every plant, but they give a good broad picture of where we are. But even within the plant, the emission factors also show high variability. Nitrogen removal is less variable, so it's kind of more constant over time, but N2O changes significantly. You can see within days how N2O emissions can change. For example, in one of the plants, the emission factor was 1.3, but the standard deviation is 1.36, which means that there's a high variation. In Switzerland, in different groups, you can see the range for emission factors between below 0.1 to a few percentage values. So there's a big range also between one plant. Why do I think it's important to account? Is because if they're not normally distributed, we probably shouldn't report them as normally distributed because we're hiding some information. So what fraction of the emissions are peaks and which fraction are baselines? Because the mitigation strategies for each one are probably going to be different. So we have a system that has a very high variability. It's probably because it has a lot of low points and some high points. And if a system has a very low variability, it's probably going to be it might be easier to find one strategy that decreases the whole mean of variability. So I think it's important to keep in mind all these values. And now, lastly, for modeling, all the information that we have from microbiology, what has been done in the last years is to translate them to equations where we include ammonia, nitrite, nitric oxide, N2O, and hydroxylamine. So the pathways that I've shown before, we just describe them mathematically. The models that I'll talk about are all ASM extensions. So they are commonly derived as extensions to ASM. So you're familiar with ASM1 or MSM2, all these commercially available from softwares. They're very, very similar. The only difference is that now we're adding 3 soluble compounds, hydroxylamine, N2O, and NO. And for example, nitritation from AOBs goes from 1 step to 4 or 5 steps. And denitrification from 1 or 2 steps to 3 and 4 steps. So we're kind of complicating models to predict N2O, but the structures are very similar. And the process rates, they're very, very similar. It's just we're extending, we're adding more processes. Now, one of the issues is if we go from 1 step to 5 steps, is that we have more parameters. And remember, we're measuring only N2O, and that's the combination of different pathways, which means that we have a lot of parameters for a new data set. Now, if you want more information about these models, this report for IBOL has a chapter on N2O production and emissions, and then describe all the N2O models that are available so far, guidelines for model selection. So a lot of the information and literature is already reported there. So I'll give some of my ideas that I have about the models, that we have to be a bit careful about the models because, as I said, we have a new data set. We have N2O liquid measurements, but we're trying to model a lot of processes. And if we think about the picture before, N2O consumption is the last step and requires 1/5 of the COD than nitrate reduction. That means that the variation in the consumption, we are not going to see them in the COD of the nitrogen balance. So an error of 1% in ammonium or nitrate or COD, that's not going to be relevant. We might not catch it in the error when we calibrate the model, but that's going to be very important, 1% in N2O. So there's a discrepancy on N2O errors and remember nitrogen species, which are orders of magnitude higher. And for example, we could have different cases where between production and consumption, the net result is the emission. But this emission could also happen from lower production and lower consumption, or from different pathway contributions and the same consumption. So to elucidate these values with just one data set is complicated. Another point is that we can describe suspended and mixed biomass, but in the models, who is really producing N2O? We've been looking at N2O pathways where we have a lot of steps, and it could be the case, what I've seen with some models, where N2O here is produced by nitrifiers, and in this case by denitrifiers. But if you look one step back, it's actually denitrifiers who are producing the substrate and nitrifiers who could be producing the substrate. So we might have this cross-mixing between substrate that is just an artifact of the model. So we have to be a bit cautious sometimes with the model because it might give a good result for N2O, but we might be missing something deep down. And this is just an opinion that these additional data sets for N2O, they don't allow for identification of pathways because we've increased the complexity a lot. So these models are typically over-parameterized. We still lack a validated model with seasonal dynamics, but we can already describe the trends. So it's very close, but I don't think we still have a model that can describe a one-year campaign. And we needed more rigorous discrimination tools because the good modeling practices that we have for water treatment, they're still not up to date for N2O yet. So a lot of calibration protocols are expert knowledge based, but not based on the information content. Or we have a lot of models that have good fits, but if a lot of models have good fit, that means that we are not discriminating rigorously. And lastly, it's pretty hard to find prediction of N2O emissions that include uncertainty. And that's going to be important to compare the quality of two different models. And for other models in the report, they also include a chapter where they talk about risk-based models, data-driven, CFD, and also biofilm models. And with that, I'll conclude. Remember, N2O is produced and consumed biologically. Net emissions can be quantified online, liquid and gas phase. Campaigns to account for seasonal variability should last months to year and check for spatial heterogeneity. And the variability in emissions, they show that they are not normally distributed and they're more skewed. So they look more like flow distributions. And we still lack standard protocols for data creation and reporting. So we're in this point where we can kind of try to develop more accurate tools. And process models exist to predict N2O production in suspended systems, but the uncertainty is not typically reported, and validation of long-term data sets still remains challenging. Thanks.
I will use the speaker. All right.
So Mikkel Andersen, he's the CTO of Unisense, and he's probably the person that I know with the most experience on N2O emissions. And he's been working with N2O liquid sensors for many years. But the most knowledgeable people on N2O. So Mikkel, whenever you want.
Well, thank you for the invitation. Yes, so my name is Mikkel Andersen, and I work as a technical director at the Unisense environment, and I've been working with nitrous oxide since 2011, developing the liquid sensor that is used in many places in conjunction with scientists and in full-scale monitoring. And what I promised Carlos was to tell a little bit about the full-scale mitigations that we've done in Denmark, and also a little bit of the background for the monitoring campaign that he also mentioned. So we know this already in terms of the emission factors and the variability that they give. Another thing that we also see, which is a real challenge that surfaced maybe 10 years ago, is that as we start to save a lot of energy for aeration, we actually lead to more nitrous oxide. So all of a sudden, the CO2 footprint of energy savings was exchanged with another CO2 footprint in terms of nitrous oxide. And these emission factors that Carlos already told you about are really not representative for what is going on in real time, and certainly not in seasonal times. And also here, this is actually the IPCC factor, so the data set behind the IPCC factor that was published in 2019. And actually all the Danish wastewater treatment plants, they are lying down here with below 3 tons of N load per day. And it's really hard to know whether the emission factor given by IPCC would be valid for Danish wastewater treatment plants as the correlation is really driven by a few very large wastewater treatment plants that have been measured. And in Denmark, it was decided simply to get more measurements. And this gets even more clear when we start to combine both the IPCC data and also the David de Haas data that was shown before by Carlos, but also now the Danish EPA data that we also saw the Danish report for. And as you can see, there's really no correlation between the loading or the removal rate of nitrogen in a wastewater treatment plant and then the emission factor that is measured. And all the blue dots here are half or more than 1 year of measurement, and the dark blue ones here are the ones that are coming from the Australian, New Zealand study, and then also the orange ones are the IPCC data that has now been checked and validated by several researchers. So you can really see that some of the bigger wastewater treatment plants that are now available are not driving the linearity in the emission factors anymore. So pick your own emission factor if you want to report something, but know that the variability will be high. What is also important is that we have seen that a lot of the new cost and energy efficient processes actually from time to time have a very high emission factors. Here are 3 Anamox type or deaminification type of reactors that are depicted. There are other processes that are much better in terms of emissions and energy efficiency that one can look at. But having nitrous oxide being measured in these technologies is certainly important. So we sell this direct sensor that allows us to measure both in the denitrification or during the anoxic zone, but also the oxic zone, and it integrates in real time into the SCADA system, and that allows us to actually get a lot of data in real time, so that we start to understand also the very fast dynamics that Carlos talked about before. One of the things that we also learned from the 2-year Danish program is really reporting the emission factor in the right way. Here with the red, and that was that factor that Carlos showed before. So the amount of emissions per load or per purified ton or kilogram of nitrogen. But one thing that is also important is to collect data in terms of how much carbon is available for CO2 for nitrates removal. And also what will become clear later is to know how much ammonium is actually turned over per sludge, gram of sludge, or per volume per time unit, because I think that will be a key factor when we want to compare different processes. The outcome of the Danish national program from the 9 wastewater treatment plants led to a new emission factor that is now implemented and used in Denmark for our national reporting. But as Carlos also showed, the variability is very high. And here, a lot of the projects that I've been involved in monitoring here in Denmark are shown on a map. And just if I take all the data here and make it a new weighted average, then the emission factor is actually slightly higher or somewhat higher than the new Danish emission factor. And one of the key things to remember is that it's actually the bigger wastewater treatment plants with a fairly high load that actually leads to the higher emission factor. So here we have in Aarhus, where I live, one of the bigger wastewater treatment plants, and we have 2 in Copenhagen with very high emission factors as well at Odense. So the major cities will all have higher emissions compared to the smaller cities around with lower load and lots of hydraulic capacity and treatment capacity. So in Denmark, all of this has now led to legislation that will point towards a mitigation or reduction of nitrous oxide by 50% starting by 2025. Simply both to monitor and know more and mitigate more once we have the knowledge that we have today. So I promised to present some full-scale data from projects that we've done. In this first case here, it's a Varga project that we're actually writing up now and will be published soon. All of the data. It's a project where we look at all resource recovery, but in particular, I've been working in the part where we try to mitigate the nitrous oxide. The project has been funded by the Danish EPA as part of a learning project in terms of what we can do once we implement the regulation. And you can see some of the partners here. And Invedan was the consultant that actually did all the collection of the national data in the report that Carla showed you before. So in this wastewater treatment plant here, we have been monitoring for more than 4 years, done 2 years of baseline, and then 2 years of mitigation strategies. It's a very long raceway plant with 2 tanks working in twins. And the red tanks up here have been used as a reference, and then the green tanks here we've been using for process controls. They are running ammonium-based aeration controllers to control DO, but also the nitrification and denitrification times are controlled in these plants in order to have an effluent quality lower than 8 milligrams per liter in total N. And one thing that becomes clear when you start to monitor 4 out of 8 tanks over time is that you start to see seasonal variability. We actually monitored the baseline in 2018, but that was a very hot summer and very dry summer here in Denmark. So we actually extended the baseline to 2019. And as you can see here on this particular plant here, we actually have very high emission factors for just the loading or the amount treated on this plant. You can also see the variability that we have throughout the year, with the summer or spring months being the most significant emitting months. And this actually continues year after year. So these are 4 years, more than 4 years of data. There are around 4.5 million data points per sensor in here. And you can see that seasonal pattern repeats year after year. But you can also see that actually the emission from year to year is actually very different when you compare them. So having dynamic emission factors, I can underwrite that by just showing this figure. So ongoing and long-term measurements are really key in order to calculate your emission factor. And it is actually, in my mind, more valid to have an online measurement system, be it one type of sensor or another, but to actually know how it's changing. And this is also the case if you look in time resolution, just and you zoom in. So this is actually a 7-day zoom in 2019 in July of the data, and you can see how the emission varies as a function of the day. But we can zoom in per day or even per hour. So you really have to choose the sensor technology that will capture the dynamics that you would like to see, because otherwise you won't be able to capture it. So we've done initial mitigation strategies in terms of trying to change the DO set point and actually changing the way that the ammonium-based aeration controller works. It's been published in 2019. And here we tried to decrease the DO set point to enable more simultaneous nitrification, denitrification. And we actually achieve a 60% mitigation. But one of the problems with a controller like this is that once we expand the volume that we aerate, then we lack the anoxic volume to decrease the nitrates, and in the end we cannot maintain the effluent quality that we would like below 8 milligrams per liter of total nitrogen. But one thing that we also learned also from literature, a slight variation of the figure that Carla showed before, is really that the specific ammonium oxidation rate is key in terms of nitrous oxide emissions. And what you can see is that the more ammonium you turn over per time unit, per amount of sludge, or as I also stated earlier, per volume, the more nitrous oxide you form. So one way to actually move down this curve would be to increase the amount of the sludge or MLSS in the tanks, and then see whether that would decrease the emissions. And we actually tried that quickly over a 1 week period, where you can see that the green ones are the ones where we increase the MLSS in the tanks, and then we have the red tanks or blue-red down here as reference. And you can just see on the concentration but also on the emissions, that with this very small change, which is just a SS set point change, we actually diminish the emissions quite significantly. So this we have actually continued to do for not only in 2020, but also in 2021. And we actually achieve reductions around, on average, around 60% between the 2 years. And then the emission factor with the reference tanks goes down and then up again due to the figure that you saw before. But if we compare with the reference tanks in the same year, then we get a significant reduction in the tanks here by just changing one set point. And you can see the changing of the emission factors here in the lower panel here in 2019 and 20 to 2021. From 19 to 22. Where we have very different emission factors. And I'll just try to compare that with a situation where we would use either one of the IPCC factors from 06 or 19, and then compare them with the old and the new Danish emission factor, and then actually the measured one. And what we often see on especially the bigger plants is that we see higher emissions when we start to measure long term, whereas these emission factor P numbers are not really working. But you can also see here that the change in emission factor and then the loading on the plant, the variability in the emission factors are much higher than the load variability here. So there's more than just the load on the plant in play when it comes to a correlation to the emission factor. And that is why we believe that the correlation that IPCC uses for emission factors is a good top-down way and approach, but it will never be very precise. And this points to the fact that the differences that we see between the tanks in this project at the this Varga project actually points to a change or a difference in the sludge between the different lines. And we do see that every year the sludge this is a principal components analysis of the DNA that has been sequenced, that we see that in high emission season, so in the early spring, we see that they are all clustering here. Unfortunately, due to COVID, we couldn't continue the sampling in the same way, so we don't have a continuation throughout the project of this. But this study has also been published here, and you can read more about it. But DNA sampling is certainly something to look into if you want to understand more in terms of the different mechanisms that Carlos showed you before. I'll skip one slide here and just go into the mechanisms that when we work with them in full scale, because this is sort of a process engineer's view of what goes on. So the pathways are here, but not the bacteria behind. So we treat and when we mitigate in full scale, we are working more with flux structure and how we can use the flux structure to change set points so that we can enable, for example, both nitrification and denitrification at the same time, but also avoiding that we have nitrates that are building up and then leading to nitrous oxide via the nitrifier and denitrification pathway. And one way to look at it is actually to see how much we load in terms of ammonia here. And if we load in too much here, then we accumulate nitrates. and then we'll end up having nitrous oxide being formed here. And this is actually nicely shown as one of the reports in the EPA report that was a part of the 2-year monitoring campaign. So here it's a plant where they have either tanks in running in parallel, or if they run them in sequence instead. So this one will have the double load compared to one of the tanks here. And you can see here from just looking at the red and the green dots that putting all the load in one tank and then having a sequential tank afterwards, taking much less ammonium in, you will have a much higher emission compared to one where you try to split or even out the load. So very similar to what we did in the project that I showed you before. We tried to dilute out the ammonium load per bacteria, where here the load is the double compared to the red one here, where we split the load between the bacteria. We also know that this COD to N ratio is really important. This is a case in mid Jutland in Denmark, where they moved from a situation without pre-treatment and then a steady COD harvest in pre-treatment, and you see how the emission factor goes up, and it actually goes up much higher than that when they start to dose chemicals as well. So COD to N ratio is important in terms of having enough carbon available for full denitrification, because otherwise it will end up as nitrous oxide as part of the denitrification. And the importance of COD we've published some years ago now, where it's a post denitrification process where we basically have no COD left for denitrification, and then dosing complex COD mixtures into the denitrification zone, post denitrification zones. And then monitoring the amount of nitrous oxide in the liquid, you can see how nicely the around COD to N ratio matches the actually stoichiometric ratio that is in literature in terms of COD to N ratio. So, am I fine on time? So this one here is a dehumidification plant where we've shown that implementing a PID controller, where we use the signal or set point of the nitrous oxide to control the amount of N2O emission. If we set the set point and use the continuous aeration in the system based on nitrous oxide measurement rather than a pH and DO set point measurement, then we can actually decrease the emission from the side stream by sixfold. And remember that I told you earlier that the emission factors on these tanks are normally quite high for side stream, but with the right tools, nitrous oxide sensor as a feedback in the control of the aeration, then you can actually also mitigate significantly the amount of nitrous oxide that is stripped up into the atmosphere. So some of the final slides, these are more technical, but I thought they would be relevant for the audience here, is really that knowing how to work with the sludge in full scale, then you can also start to do more advanced process controls. This is data from a pilot plant, 2 times 1 cubic meter tanks running a classical recirculation mode. And what you can see is if we lower the oxygen enough, then we are actually able to remove both ammonium while oxygen is available, but still have a significant removal of nitrate as well. In this case here, it's a nitrate measured, but also the nitrite is very low. So actually what we are trying to achieve here is to have a situation where we can have simultaneous nitrification and denitrification. And in models, this is really hard to capture because these are calculating on free bacteria. But here we utilize the diffusion gradients that we have in the flux structure, and we saw a beautiful picture of a granular earlier in the introduction. With you saw the AOBs and the heterotrophs are separated in a 3D space, and oxygen and nutrients will of course diffuse into the granular and have gradients. And in these gradients, then we will be able to actually do several things at the same time in one particular flock. And using this information and plotting all the data, it's a very messy slide, but that actually gives you some hint to what you can do in terms of removing or nitrous oxide. So a lot of you have probably heard that if we decrease the oxygen enough, around 0.5, then we will see an increase in nitrous oxide. And that is also very true. And one of the rule of thumbs is really to have a high enough oxygen to avoid the nitrification, denitrification pathway running. But if you then continue to lower the amount of oxygen, then you're actually ending up in a situation where you can have a significant sink of nitrous oxide while still turning over some ammonium. Not a lot, but enough ammonium to actually have a significant ammonium removal. And the only thing that now regulates the amount of nitrous oxide being formed is whether you have COD available or not. So what we have tried to do in several plants now is to really find this sweet spot where we can be somewhere around here, where we are sure that we have enough COD available to avoid nitrous oxide formation. So here's the rate that is plotted. So dosing or controlling your COD level while also having a really fine-tuned level of oxygen in the liquid, you will end up having a really nice NDN or shunt method. We actually don't know whether we have a shunt, so where we are not creating nitrate, but rather nitrite. So in order to have a lower emission. And this we also shown in the pilot scale here, where we changed from a typical alternating system with the aeration on and off over 14 days, and then we changed it into a recirculation plant, and then slowly lowering the amount of oxygen in the bulk liquid, so adapting the slots to a simultaneous nitrification denitrification situation. And I should say that the effluent quality here is still perfect. It's below TN of 8, and the COD is completely used now for denitrification as part of this, and with low slots production. So here you can see that the nitrous oxide decreases, and we actually below the detection limit of our sensor in this case here, and the oxygen level is around 0.2, 0.3 milligrams per liter in this case here. So we save both on energy and on emission at the same time. These controllers are not easy to do, but they do work, but they require that you have 1 minute data to work with, and then actuators in the wastewater treatment plant that are fast and responsive in order to get it to work. And this part here we have actually now implemented together with Environmental at a utility here in Denmark, and it's running on 4 wastewater treatment plants now. And one of the things that we actually gain by implementing a control like this is that we actually double or more than double the capacity of the wastewater treatment plant while doing this. We still need controllers to capture, high load rainwater and so, but the majority of the time we can run with controllers like this in real time and full scale. So summing up, we have together with a lot of our sensor customers been able to reduce both in the side stream and in the mainstream with some of the controller companies or companies that sell controllers, but also with wastewater utilities directly, we've been able to implement the mitigation and reduction strategies that are quite effective. And just as a side note, the cost effectiveness is really clear. So if we do get a carbon tax on CO2, then we offer, together with partner solutions, where we can remove a ton of CO2 from 4 to 30 or 40 euros per ton. And right now, a ton of CO2 is trading around 100 euros and is expected to increase to 200 euros per ton by mid of this decade. So this is the last slide, and maybe just for the audience here, the stepping stone to become climate neutral, really to introduce equipment that is controllable and energy efficient. But the next thing then is to do mitigation of process emissions. They are the biggest part of the carbon footprint from the water sector, and only then start to do sustainable energy production, and not just biogas, because the grid will be green very soon, but produce something that can actually be used to displace other carbon sources like petrol or even produce hydrogen. So with this, thank you for letting me present.
So my name is Bing Guo. I'm a lecturer at the University of Surrey. I study environmental biotechnology, microbiology, and I'm happy to be one member of the working group in the Unisense nitrous oxide from environmental biotechnology. And this is our first webinar, and I can see it goes smoothly, goes very well. We have a lot of audience online, even though there's short notice. So we will have more events coming in the future, and do subscribe to EBNet and get notifications of other future events. All right. Okay, so let's now look at the questions. So the first question is about simpler tests on site. There is a question that on site in the treatment works, they can test gram and nasal staining easily with their equipment on site, but not DNA sequencing. So do we have a way to link the nitrous oxide emission with simple methods that the on-site engineers can do easily? Yeah, Tom, maybe.
You just talked, I'm just talking.
Just go for this mic here. Okay, so basically no. And it's not possible, and it's not intrinsically possible. And I think really that's sort of the bad news. The good news is that DNA sequencing is getting very cheap and very portable, and there's actually no reason in principle why we cannot monitor the abundance of the relevant genes for the ammonia oxidizers, the different classes of denitrifiers, certainly frequently, and if we wished, it could be done daily or almost in real time. And I think really, when I'm talking about the opportunities, I think this is an opportunity for the industry to start to adopt some better techniques, and that will help them take better decisions. So the portable DNA sequencing is a fact now. It's something we do fairly routinely. In Newcastle, we know that it's been done fairly routinely in Denmark and quite a few other places. But it's the nitrous, for example, has it's very good for telling you something about what's been accumulated in the cells, but it doesn't really tell you anything about the abundance of the key genes, and it's not intrinsically capable of doing it. So I think for some of these things, we've got to move on and embrace some of the newer technologies. They're not that expensive. It's more like I think a training and dissemination issue rather than a research issue.
Thank you. Yeah. Thank you. I think following that, so there's a comment from Sonia also saying that we need interaction between system biology and engineering, process engineering, or maybe the engineering education, and hopefully maybe in the future we can have some workshop or training sessions, to bridge the gap between the more advanced technologies and on-site access. And the next question, is there any known research on nitric oxide dismutation in wastewater treatment? This metabolic phenomenon has been observed in anoxic deep sea environment. Can it be relevant to wastewater treatment plants?
So as far as I know, it's not a very relevant pathway, but I know like people were trying to investigate also in lab scale if that might become relevant. If it could be a pathway, the problem because it's also sharing intermediates to elucidate whether it's significant is quite complicated. If it's because sometimes they were thinking like maybe releasing oxygen might also be affecting, that could have an effect on N2O production. But as far as I know, in current literature, it's not considered important. Okay.
Thank you. And the next question about factor N simulation into biomass, I think Carlos answered that in the chat box. And then the next one from Sonia, what do you consider to be the biggest limiting factor, data size or intrinsic variability or actual model parameterization, sophistication?
I think that with ASM models, which are the kind of the more, or most people were working at, because they've been so over-parameterized, data set is not going to change. So N2O is going to be only one data set, and that's not going to change. And you cannot identify 8 parameters, new parameters from there. So most of them are fixed, and the model is not sensitive to those. Some pathways are, but so I think they're overly complex, but I think it got to a point where we had to complicate them as much as possible to gain understanding. Now, maybe we can try to simplify them a bit more because we know what's sensitive and what's not. And also, of course, at the beginning, data sets were not very good, and now we have very, as Mikkel was showing, very long-term data sets with very good inputs also for other variables that before we didn't know could be important. So data sets are getting better, and the models could get simpler, I think that would be. But it might be that we get to a point where mechanistic models are not important, and we can go for data-driven if the data set is good enough.
Thank you. Any other thoughts from the working group? The speakers? No. Okay, we all agree that.
I've got one question myself.
Yeah.
So you, Carlos. Well, you mentioned. Yeah.
Put it in the chat and we'll get it then.
To the chat and speak out.
Ask another question on this.
Yeah. So we have a couple of process engineering related questions actually. For example, the next one asks, if you increase the plant's hydraulic load capacity, can you assume by default you will reduce the production of nitrous oxide? Mikkel, are you online? Can you maybe?
Yes, but I cannot hear the question, so I was just trying to find it in the chat as well. But, can you repeat the question, please? It's muted now.
Hydraulic loading capacity, does that lead to changes of the nitrous oxide production?
In terms of the hydraulic loading capacity? Well, it's actually more the treatment capacity that will lead to higher emission factors. So as city grows around the plant, we get better and better at getting rainwater out of the combined sewer system to actually increase or maintain the hydraulic or maintain being below the maximum hydraulic capacity of the city. then the load and the strength of the wastewater will increase, and that will lead to higher emissions. In wet weather, we do see high nitrous oxide at the very beginning, but then due to the higher load of the first flush, so to say, but then you can actually have long periods afterwards with low nitrous oxide because the wastewater strength is lower. But that is more dictated by the sewer system structure and delays than it is in terms of the processes that go on the wastewater treatment plant. I hope that was. I only heard half the question.
Yes. Yeah. Okay. Sorry, there's still switching on and off the microphone. Yeah, I think that answers the question. And I think the next question, maybe Mikkel can also give some insights. Okay, there's one comment saying like because the seasonal temporal variation in your presentation saw some increase in the years of 2020 to 2021. Is it something related with COVID, like the life pattern that people discharge, or maybe?
That is a very good question. That is actually one of the things in doing research or trying to do mitigation research in full scale is that what you thought would be an excellent reference that you kept alone starts to change and differ from the tank that you're working in. So what we have done actually over the last during the full period is that we have looked at all the sensor input on all the 4 tanks or all 8 tanks in the wastewater treatment plant, and we've shown that they are completely the same. If you accumulate all the data over 4 years, so 4.5 million data points, all the tanks are more or less looking the same with very little change. They have also just concluded a sampling on the run into every of the tanks, and there's no difference in the volume or content of the wastewater that runs into the tanks. So that leads to the thing with the DNA, so that the slots in the different lines that isn't shared between the tanks is different in the tank where we change the MLSS compared to the reference tank. So this is, we only have that PCA analysis that I just showed you as the only true signal. The rest of the data are all the same. So when it comes to modeling these processes or even doing machine learning on these, how do we actually collect and capture data that will actually capture this difference as well? There's some uncertainty that we will inject into the systems by doing this. And it's also a challenge on sensor-based controlling, of course, because the nitrous oxide measured in the tanks are different.
Okay, thank you. Actually, the next question is related with controlling. So Ben asked, what happens to...
It's already been answered.
Oh,
okay.
Yeah.
Okay, sorry. I'm not... Then... Sonia asked, I think this was during Mikkel's presentation. What is the control on the second option?
Yeah.
Just the flat concentration.
Yeah, so that was the dehumidification. And so in this case here, a classical dehumidification is made by controlling the pH, which is a very efficient and well-proven way to control it. One of the things with the change in pH, but also the amount of time that you have air on, that leads to an accumulation of nitrite in the tank. And the higher nitrite, the higher the emission of nitrous oxide will be. So the philosophy behind the controller was really, if we can keep the nitrous oxide down, then we know that the nitrite is low. So having a PID controller based on monitoring nitrous oxide and then feeding that back into the aeration control, then we could have a PID controlled aeration control based on the NMOX activity rather than on the ammonium or AOB activity.
Thank you.
Yes.
The next question is also about kind of like process control. If a plant keeps control of its FM ratio, can we assume that nitrous oxide is kept at a minimum?
Yeah, again, a good question, because you can have a good sort of COD to N or food to active bacteria ratio. But if the N load is too high, so again, if you turn over too much ammonium in a short period of time, then you will still have high emissions. The COD is important. If you do full denitrification, so full BNR, then you'll need that food ratio in order to sustain an efficient denitrification. I'm not so familiar with the ins and out of the F to M ratio in terms of the regulation, because it's not used that much in my part of the world.
Thank you. Audience asked about the presentation, and also some document links has been sent to the chat box. We have recorded this session, and we'll check with project partners of what information can be shared, or 100% can be shared, and then we'll upload the online version, online recorded video, so the audience can come back and watch.
My slides can be shared, and so I think.
Okay, that's great. Thank you.
Yes, I'll PDF them for you.
Next question is to Carlos. You mentioned using mass transfer coefficient as a method for estimating nitrous oxide emission. Is this something you have investigated, and if yes, how do you validate this method?
There's like extensive research, so there's different methods. Like in the kind of more basic, if you have the mass transfer coefficient for oxygen, which some plants might have just for the oxygen transfer efficiency, there's some very simple correlation with diffusivity, where you can compare the KLA for DO to KLA of N2O. If you could have direct measurements, so liquid N2O measurements and gas N2O measurements, you can already get the KLA value directly. But if not, there's different methods. So also with dynamic oxygen profiles, you can get KLA for oxygen, and you can convert them back to N2O. So there are different methods that are reported that are available.
And if I can comment as well, so also note that there's a difference between whether you are working with surface aerated or bottom aerated systems, because the bottom aerated systems are more driven by an equilibrium term, whereas the surface aerators are driven by a sort of a KLA based term. And we have shown and published that the mass transfer part, so the KLA impact on the emission calculations based on bottom aerated is not impacted that much, but still it is an empirical formula that is used today for KLA estimations, as Carlos also mentioned in his introduction.
Okay, the next question for Mikkel. Thank you for the presentation. During the trial, apart from the process parameters being measured, were there any measurements of mixing or turbulence, the physical releasing? I guess there's like, what is there a variable, like changing parameter monitored?
Well, all they are quite well equipped, the treatment plant. So we have all measurements in terms of. There's an echo here from. There's we measure both airflow and inflow, influent flow, and all sort of the parameters, including nitrate and nitrous oxide, oxygen, and ammonia. And no parameters were really changed in the plant during actually the 4 years, except for the changes that we made in the controlled tanks. So the puzzle is really to figure out why are the tanks different when the measurements are the same.
Thank you. I think we have the last question in the chat box here. Does increasing the alpha factor have a positive effect on nitrous oxide emission?
Yes.
Yeah, we've seen that with a model, but yeah, typically, yeah.
Yes. More efficient stripping.
Okay. All right. Okay, another question. What was the maintenance requirement for keeping all the key sensors working accurately?
For our part, actually, our sensors are fairly maintenance free. They need calibrations every now and then, so bi-monthly or every month, and regular cleaning. But they don't foul like other sensors. I showed a picture of the tip, and it's because it's so pointed that the water will actually, with shear force, polish the tip itself. But like all other sensors in wastewater treatment plants, they are regularly looked at and cleaned for rags and also calibrated. But they are certainly on par with any other sensor, including the DO sensor. But running advanced process controls rely on sensors that are actually working. So sensor maintenance should be an important part of the control strategy if you want to implement more advanced process controls. And I assume some in the audience are already running ammonium-based aeration controllers, so they would know that maintaining the ammonium sensor is, of course, critical for having a good and energy-saving ammonium-based aeration control.
Thank you. And I don't see more questions in this great session. Thank you again for the speakers, and thanks the audience to coming to this online session, and hopefully to see you in the future, either online or in person. Again, the documented recording will be checked and shared with the public. So you can register for your membership on ebnet.ac.uk or send the email to ebnet@ebnet.ac.uk. So I'll let the EBNet managers to share the links and the documents. Thank you, everyone.
How are you doing?